Hello, my name is Chloe Olson. I'm going to be talking about replication. So replication is the process in which filamentous bacteriophages reproduce within their host cell. It replicates through a rolling circle mechanism, and the steps included are attachment and entry, conversion to double-stranded DNA, viral genome, replication, packaging, phage, assembly, and release. The replication process is in the attachment and entry phase. The phage attaches to specific receptors on the surface of a host bacterium, but they do not inject their DNA into the host. Instead, they bind to the receptor of the viral genome, the single-strand DNA, and enter through the cell's pilus. Next is conversion to double-strand DNA. Once the phage is inside the bacterial cytoplasm, the single-strand DNA genome is converted into a double-strand DNA from the host's replication machinery. The now double-stranded DNA is used as a template for further replication. Next is the viral genome replication. Using the rolling circle method, a DNA polymerase starts synthesizing the complementary strand by creating a long polymer chain of viral DNA. The method is initiated by replication proteins encoded by the phage genome. The next phase is packaging of a genome. The newly synthesized single-strand DNA genomes all have protein codes to form a protective capsid structure as they are transported to the cell membrane. The last step is the phage assembly and release, where the single-strand DNA genomes are inserted into new phage particles that are assembled at the bacterial membrane. These filamentous phages are extruded one by one through the bacteria's cell membrane. The phages do not immediately kill their host as lysocytic phages do. Instead, the host bacteria survives and continues to replicate, and the newly formed phage particles are released into the environment to infect a new host.
In the filamentous phage, there are bacteriophages that consist of long and thin bacteriophages, which infect bacteria and archaea, and they usually target E. coli, which is one of the main bacterial cells that they target. And they're part of the Inoviridae family. And the structure of this phage is made up to thousands of protein subunits forming the helix around the DNA. And in this phage, they replicate and exit their host without killing the cell, which means that they can be applied to biotechnology. So for example, the biotechnology that they use is phage display technology, which helps them study protein interactions, and then they can also develop new drugs. Moving on in the filamentous phage, there's a cycle that applies to this phage. And there's 5 components in the cycle. One of them is attachment. This phage attaches to the bacterial cell due to specific receptors, such as proteins and lipopolysaccharides, on the outer membrane. And DNA injection is where phage DNA is injected to the bacterial cell through a pilus. Then in the replication part is where phage DNA is replicated and produces a completely new RNA, which leads to assembly, where the host cell is produced into new phage particles, and then new phages are made and expelled out of the cell, which leads to our last component of the filamentous phage cycle is release. During the release, a new phage is released. That's it. So in the filamentous phage cycle, there's these 5 components: attachment, DNA interjection, replication, assembly, and release. And this is what happens in that cycle.
Generalized transduction is a type of horizontal gene transfer. In generalized transduction, bacteriophages accidentally package host bacterial DNA instead of its own viral DNA during the assembly of new phage particles. This error creates what is called a transducing particle, which carries no phage DNA and therefore is not a phage. Due to the accidental packaging of the host bacterial DNA, generalized transduction now has the ability to transfer any segment of the host DNA. This process of generalized transduction starts with the phage attachment to the host. Then the phage DNA enters the cell. Following this, enzymes that are encoded by the phage cut the bacterial DNA, and then nucleic acid is replicated while proteins are synthesized. Lastly, during this process, during the construction of viral particles, bacterial DNA can mistakenly enter a protein coat, and that is generalized transduction.
Specialized transduction is different from generalized transduction because it only transfers specific DNA sequences located near the prophage integration site. And in specialized transduction, it's a form of horizontal gene transfer in which bacteriophage transfers specific bacterial genes from one host to another. And this occurs when a prophage exits the cell incorrectly during the induction process, and it carries the adjacent bacterial DNA along with it. And so the steps for transduction include integration, which is when a bacteriophage integrates its DNA into a specific site of the chromosome of the bacterial host, and then it becomes a prophage. The host bacterium replicates and divides, copying both its own DNA and the phage DNA. And then the second step is when the prophage exits the bacterial host, taking some of the host's genetic information along with it. And this is the key step that differentiates specialized transduction from generalized transduction. And then in the third step, this is when the newly formed viral particles begin to assemble, carrying DNA from the previous host. And the phages cannot carry out a full viral replication cycle since they only contain bacterial genetic content. And then the fourth step is the infection of the new host, when the phage injects the hybrid DNA into a new bacterial host. But because the viral DNA is incomplete, a full infection does not occur. And in the last stage, after the defective phage injects its hybrid DNA into a new bacterial host, the bacterial genes from the previous host do not degrade. Instead, they integrate into the new host's chromosome through a process called recombination.
Hey y'all, I'm going to be talking about the different strategies that bacteria use to defend against phages. So basically what a phage is a virus that can infect and replicate inside bacterial cells. So first we have surface modification, where bacteria can change or mask their surface receptors, which are the entry points for phages. By doing this, they effectively block the virus from attaching and initiating infection. Next, we have the CRISPR-Cas system, which is basically adaptive immunity, where bacteria can capture and store fragments of phage DNA in their genome, and then when that same phage attacks again, the system recognizes the DNA and targets it for destruction. And then we have restriction modification systems, which use restriction enzymes to cut up invading phage DNA. Meanwhile, bacteria DNA is protected from these enzymes through methylation, which acts as a molecular shield. In abortive infection, infected bacterial cells initiate a self-destruct response, which kills the cell but stops the phage from replicating and infecting neighboring bacteria, protecting the larger population of bacteria. And then prophage immunity involves phage DNA already integrated into the bacterial genome. This prophage prevents other similar phages from infecting the same cell, often by repressing the replication. And then lastly, we have emerging mechanisms, which include discoveries like the antitoxin systems, specialized antiphage RNAs, and the Zoria system. These novel defenses are broadening our understanding of bacterial survival tactics. So yeah, each of these strategies highlight how bacteria continuously evolve to counteract phage threats.
The Bible calls people to love and to serve others, which can be done by providing proper care to patients and helping them through their struggles. Medicine is viewed as a gift from God that healthcare personnel can use to help heal patients. 1 Corinthians 10:31 says, "So whether you eat or drink or whatever you do, do all to the glory of God." This is a good reminder for us that throughout our daily lives and our careers that we should be doing things to glorify Him rather than ourselves.